Um, Hi, so I'm here to talk about the BioCyc Web Services API. I'm going to begin with a very brief description of BioCyc because I'm assuming that most people who are interested in this might already be familiar with BioCyc. And then I'm going to just launch into the different kinds of services that we have available. And there's kind of a lot, so I'm not quite sure how much detail I will go into, but we'll see how it goes. Um, so BioCyc is it's our website. It's a collection of over 20,000 what we call pathway genome databases. We have uh, a pathway genome database is collects. We call that because it collects information both about or all about the genes and replicons, et cetera, in an organism, as well as the enzymes, the transporters, the pathways, the reactions, and it's got various other things like Go terms, et cetera. We have a couple of highly curated databases. EcoCyc for E. coli is our sort of flagship database, and that's the one that most of my examples today are going to be taken from. We also have MetaCyc, which is not an organism database, but is a database that takes information from all organisms purely on metabolism. It doesn't contain genomes, etc. And then we have a collection of decently curated databases for organisms that are of interest to large communities, like tuberculosis and Bacillus subtilis. And we keep adding new ones to this collection as we curate them. And then the vast majority of databases are purely computationally derived based on our own pipeline. So there, it doesn't contain as much citations back to the original literature. Well, much is not known about these anyway. So BioCyc is the software that underlies BioCyc is a package called Pathway Tools, which we also develop and which can also be installed and licensed separately. But and BioCyc runs on this. So between BioCyc and Pathway Tools, I'm not going to go through this entire list, but we have a wide range of capabilities for interacting with the genome and genes, various ways of exploring pathways, regulatory information, various omics analysis capabilities, comparative analysis capabilities, and then, various other things. I'm not going to go through this. What we're here to talk about is web services. And so what are the use cases in which someone would use web services with BioCyc? The first one that one could think of is if you want to retrieve data programmatically. So this is if you're developing like an application that wants to retrieve, say, a small amount of data at a time on demand. In particular, what we do not want you to do is to use the web services for bulk data downloads. And we provide other options for that, mostly because that can that puts a load on our servers that we do not appreciate and can impact performance for other users. So instead of getting bulk data downloads, we have smart tables, which I'll actually go into a bit later because there's also web services related to smart tables. But you can export smart tables of large amounts of data, and we also make flat files available. So please don't use web services for that. But in XML or JSON or text format, depending on the service. Most of them are available in XML. Some of them are additionally available in these other formats. So you can retrieve data that way. You can retrieve images, pathway images or compound images. And then you can, this is not so much retrieving data as uploading data to our own pages, for example, to paint omics data on pathway diagrams or the metabolic overview diagram. And then you can manipulate our smart tables with web services. And so we will touch on all of these today. We have a document that is that lists all our web services. If you go to our website and look in the little footer section at the bottom, there's a link to the web services document, and that is going to list everything that I'm going to talk about today, and more with more details. And if you've looked at it in the past, I pretty much just overhauled that document in preparation for the webinar today, so it may have changed, or it has changed. I just want to mention we do require people to have BioCyc accounts in order to access BioCyc and to log into those accounts. Creating an account is free. If you don't have an account, you'll find things work initially, and then after your few non-logged in accesses, suddenly stuff will stop working, and you will wonder why. So you need to create an account, log into your account. We also require a subscription to access premium content, which is all databases other than EcoCyc and MetaCyc. Many institutions already have subscriptions, but even if your institution has a subscription, you still need to create an account. If you sign up for an account, you get 1 month of free access. And as this image shows, you can look at your banner when you're on the BioCyc website to see if you're an institutional subscriber and to see if you're logged into your account. As I said, we have 2 free databases. The subscriptions support curation of all our other databases. So when I said that we have an increasing number of what we call tier 2 moderately curated databases, that's where all the subscription revenue goes. And we have here a little coupon if you actually want to subscribe. Let me get that out of the way. But the point of all that is, if you're going to access the web services, you do need to provide your account information. So what you need to do is you need to establish a login session. We will send back a session cookie, and then you will just need to pass that session cookie with all your subsequent requests. The web services document gives examples of how to do that using both curl and Python, and hopefully you can whatever other language you're using, you can extend to that. Right here is an example of how one might do that using curl. So here we're posting the email and the password to the account to this address, and we're saying save the cookies in this cookies file. And then for every one of our other requests that we issue to the same host, we pass in the cookies file. And it's as simple as that. So let's get started talking about our data retrieval web services. We have both some general data retrieval web services, and we have some more specific ones. The general services work for pretty much any object type. So when I talk about objects, I'm thinking all the way from genes, compounds, pathways, reactions, as well as various, the objects that we use to represent, regulation and catalysis and everything in our schema. So each of the data retrieval queries is for a single organism database, or a single database. MetaCyc counts as a single database, even though it covers multiple organisms. And it allows you to retrieve data for one or a collection of BioCyc objects using our own XML format. So you can retrieve by specifying an object by specifying its identifier or its name or accession identifier, or for a compound, chemical formula, etc. So that's retrieving a single object. You can issue a query and retrieve data for a collection of objects that satisfy the query. You can also issue a general query using our BioVela query language, which I'm actually not going to go into today because it's a little bit beyond the scope here, but I will talk about the other ones. What it does do is it requires some understanding of the BioCyc schema, because if you're going to be able to interpret the results that you get, a lot of times data is spread across multiple object types and may require multiple queries to different types of objects. So we have a schema guide that is at this link here, and this is the overall schematic of the kinds of objects we have and their interrelationships. So I'm not going to go through this completely, but we have, for example, genes. which are linked to their products, which can either be polypeptides or RNAs, and of course their inverse links back. Genes linked to the replicons that they're on, as well as the transcription units or operons that they're a part of. Proteins or polypeptides can be parts of complexes, and both polypeptides and protein complexes can be enzymes. So what they do is they link to this intermediate object that we call an enzymatic reaction, which is used for linking one protein to one reaction and contains information specific to that linkage. And so the enzymatic reaction object links to the reactions, and then reactions link to both the compounds that are on its substrate, or its reactants and its products, and also to the pathways that are involved in. And this is not a complete list of our relationships because we have all sorts of things, but these are some of the important ones that you may need to understand if you want to navigate the output. We also have various information about regulation of transcription units here with promoters and binding sites and transcription factors. So I don't expect you to understand the schema now, but this is a reference that you can look at, and there's more information at the link that I gave you. So again, I'm not going to go through, this is an example of p-tools XML for a single object. I'm just going to show you a few key things. This is for a single gene. So this whole thing is, this is the gene, it's E. coli, which is our E. coli database. And this is the object identifier. It's got attributes for the name. You can see this is THRC. It also has, so this is various information about the gene. It has links to its product, which is where a lot more information is stored. And its product is a protein. And what it tells you here is, this is the information you need to retrieve information about the product object. So it's from the same organism. Its identifier is this 3-syn monomer. And so this is actually the, this resource link is the link that you would use to get information about this particular object. I guess I should also point out that we have, amongst various other attributes, the position of the gene, etc. And we have links to other databases. So this is a link to one other database with the URL that you use to link it, the ID, etc. Let me just show you a snippet. This one was, we have multiple detail levels for our XML. This was actually the full detail for the gene, although I've hidden a bunch of stuff in these ellipses here. This I have navigated to the, this is the get XML for this gene product here that we were pointed to. And this is actually the low detail version because the full detail version wouldn't fit on this slide in any readable fashion. But I just wanted to point out here that this links back to the gene, as you might expect, by the gene attribute. So this is a link now how to get the gene. It also links to the enzymatic reactions that this protein catalyzes. So this is a reaction identifier. At low detail. But I want to point out, so we had these resource links that show the links back to other objects that you need to retrieve. There are also resource links to things that appear in this document. So in this case, this is a link back to itself. So the enzymatic reaction links both the protein and the reaction. So it has links both to the protein and the reaction. Well, we happen to be looking at the page for the protein. So instead of doing this get XML thing, it just says, it's here in this document under this identifier. So you just need to find that identifier to get information. So those are the two kinds of resource links that we have within our XML. And there's various other information here, and most of it is hidden. I'm not going to go into detail more about the p-tools XML because it's reading XML is boring, and I don't want to lose you all. But it should be fairly intuitive, I hope, if you have some understanding of the schema, and we have a document that describes it. So how to actually use our web services to retrieve these objects. The basic one is retrieving it by object identifier. And we showed some of those links within the XML that I just showed you. So the basic format is this get XML. You need there's two versions of it. You can just have the ID, which is the org ID colon and the object ID. Or if you want to actually specify the detail level, you can say the ID equals this, and then the detail level is, and we offer 3 different detail levels. The none is just the object identifier, which is not useful if you just passing in the object identifier, but it is useful if perhaps you are issuing a query that returns multiple objects and you just want the identifiers. So the organism identifier identifies the organism database. So some of these are, some of our older ones are very understandable, like E. coli is E. coli, meta is metabase. Most of our recent ones are these more unreadable GCF numbers, but they all link to a single database. And then you need the object ID for the identifier for the object. This could be a pathway or a gene or a compound or whatever, and it's case sensitive. So the detail level, the full lists all the attributes. That's the default when using the get XML query. The low detail is the one, essentially we've predetermined certain attributes that we consider and relationships that we consider important to include. So that's what you get in low detail. And then none, well, you just get the ID. If you don't know what are the lists of org IDs to put in here, you can get a full list of our org IDs using this query, and it will return XML, this sort of PGDB object for every PGDB, all 20,000 of them. It will list the org ID and then the species, the subspecies, strain, etc. So that's how you can get the org IDs. If you don't know the object identifier, then you have to search for it. And there are different ways to search for it. But one of the common ways to search for it by name or accession. So this is this, and searching is always in a single organism database. So you need to include the org ID, it's this name search, and then you need to provide the object, which is the name, and then a class. So you have to specify what class you want to search. The classes, for example, genes, proteins, compounds, pathways, reactions. So all those that we looked on the diagram of the schema, all of those object names. Those are the classes. So you can search within one of those. The name, as I said, is the name or accession of the object to retrieve. We're searching exact matches only, but we search for a variety of names. For example, for a gene, we might look at the common name, the accessions, or one of its synonyms. So here's some examples. The search is case insensitive. So by default, you'll get you get output here in sort of tab delimited text format, which will list the ID and the common name for each of the matching objects if there's more than one. You can also specify that you want the result in either JSON or XML format by using one of these other invocations. When you get it in XML, you get your ptools XML format, which gives you full detail level for, but by default, for all matching objects, if there's more than one, or the one matching objects. But you can also supply the detail argument if you do not want that. Another common way of searching for an object is if you have its ID from some other database. For example, if you have a UniProt ID, and you want to search for the protein by that, or if you have a KEGG ID and you're searching for the compound. So we have this foreign ID search, and this can actually, you can actually pass it multiple IDs and it will return you the matching objects for all of them, all of them that are found. So we have, our BioCyc databases contain a lot of links to various other databases. We do not contain complete linkage to other databases. So just because you can't find something under some ID doesn't mean that we don't have that object in our database. It just means we don't have that object with a link to your database. But we do have a lot of links. So again, you need to specify which organism you're looking at. You need an identifier for the external databases. Example, KEGG, UniProt, PubChem. I'm not sure where we have a full list of the databases, but I can find that out if people need to know that. And then you need the ID in the external database. Again, the default output is going to be tab delimited, where if you supply multiple IDs, it will list for each external ID whether or not a match was found, and if so, what is the BioCyc ID. So here is, this one is an example where you're supplying multiple IDs, comma separated. And then we have JSON and Petals XML format. The JSON format also indicates for each of the IDs you specify whether something was found and what the match was. The Petals XML format doesn't actually do that. It just returns the full description of all the matching objects. But of course, all the objects contain their database links. So you could see if you could find which links match which thing there. So I'm just giving some examples here. This is the get XML to retrieve objects by our internal identifier. So this is the example where it's just the org ID and the object ID. And here we're specifying it as a slightly different way, including the detail parameter. This is an example of name search. Sorry for the changing fonts. I wanted to get each one on a single line. But so here we're specifying a name, and it's a gene. Here we're specifying an accession, also a gene, and we want our return value in JSON format. Here we're retrieving a compound and in XML format. And then here's some examples of the foreign ID search. Looking for a single KEGG compound or a couple of different IDs from two different databases. Requesting a UniProt link in JSON format. So just some different examples of how this might be called. So those are for retrieving a single object or objects, or you're searching for a single object. You can also retrieve objects using a set of API functions that we provide. So in addition to in addition to the BioCyc website and the web services, the Pathway Tools software that underlies BioCyc contains a set of API functions for people to interact programmatically with the data. And so what we've done is to take a subset of those API functions and make them available via web services for retrieving objects. And these are based on relationships between objects. So for example, you can use them to navigate the class hierarchy. So there are functions like get class all instances, which given a class, say pathways, returns all the objects in that class. Get class all subs returns all the subclasses of a class. And so you can navigate and the supers gets the supers. There's the all subs versus the direct subs. So these are different ways to navigate the class hierarchy. We have a whole other set of functions that allow you to follow the different relationship links that aren't sort of necessarily obvious. So for example, if you wanted to get all the pathways that a gene is, all the genes of a pathway, and you wanted to do that by following individual links, by fetching the frames and then following the appropriate links from a pathway, you would need to go to the you'd follow the link to the reactions of the pathway, and from there, the enzymatic reaction objects, and from the enzymatic reaction objects, you would need to follow to the protein, and from the proteins, you'd need to follow, perhaps it's a complex, so you would need to go to the components, and from there you get to the genes. Well, rather that's a lot of work to go through, so instead we've created this API function, which allows you to supply a pathway and just get all the genes. And it's reciprocal. Given a gene, you can get all the pathways, etc. And so we have a whole set of these kinds of relationships that just make it easier to navigate the hierarchy. And then we have a few API functions that encode even more complex logic. So, for example, the genes regulating the gene, there's a whole a whole slew of different ways that a gene can regulate another gene, a gene product, really. You can regulate transcription, translation. It can regulate the enzymatic activity, and there are different ways it can regulate transcription. For example, it can, well, I'm not going to go into it all, but there's a lot of complex logic in here, and so we encapsulate it in these API functions for finding genes that regulate genes and genes regulated by other genes. So this is just representative of the kinds of relationships that the API functions we provide. The full list is in our web services document. So the way one does this is we use the API XML service, and you specify what function you're calling, and then you also need the object that you're starting with. For example, if you're getting the genes of a pathway, your starting object needs to be a pathway in a particular organism. And again, we have the detail arguments. So the result is going to be an XML document that contains the complete set of objects that match the query. So here are some examples. We're getting all the genes of the glycolysis pathway in EcoCyc. So we specify the function as genes of pathway, and the ID of the object is its org ID, and then its object ID. This example is getting the pathways of a compound in BioCyc, the genes regulated by the CRP gene, which has this identifier in EcoCyc. And here this is actually a global regulator. There are a large number of regulated genes. So I've asked for no detail. I just want the IDs of them all. And then this is an example interrogating our class hierarchy, where we start with the BioCyc pathways class, and we want the direct subs of it. And then we can, if we want to build up a model of the BioCyc pathway hierarchy in BioCyc, then we can, once we get the direct subs, we can look at each of those and ask for those which are their direct subs, etc. And so that way we can navigate the hierarchy. So that's it for the sort of the more generic searches that can cover large numbers of things. We also have a larger set of searches that are specific to compounds, the different ways that you can search them. And these can accept multiple values. So comma separated, and you can ask, excuse me, the output can either be in tab delimited format, JSON format, or XML format. So you can retrieve objects by chemical formula. That's what CF is, and then you would list one or more chemical formulas, comma separated. So this is just an example here. If you're doing, if you're coming with mass spec data and you want to find the all the compounds that match some monoisotopic molecular weight, we have a special service for that, where you list one or more weights, and then the tolerance, which is how close you need to be to that weight, and then we will return a list of matching compounds in BioCyc here, or whatever organism database you specify. You can retrieve compounds by INCHI, or you can retrieve compounds by the chemical structure search by SMILES. We also have a metabolite translation service, where you can search by multiple criteria. And I don't have an example of that. It's you can see a description of it in our service document. But essentially, this is for if I have a bunch of IDs and I don't know which one of them is going to match something in the BioCyc database. So let me provide maybe a KEG ID, maybe a PubChem ID, maybe a name, and hopefully one of them will match. And if one does, we will get that back to you. So those are the specialized compound searches we offer. The other data retrieval service we offer is BioPACS pathway data. So BioPACS is a standard language it's an XML format language for exchange of biological pathway data. And our group was actually involved in helping develop the BioPACS standard. So we've had a lot of input to it originally. We're not so involved anymore, but I'm not sure it's still changing anymore. But it's an XML format using OWL, which is it's a logic description language. And you can retrieve any pathway from BioCyc in BioPACS format, which is very different than our own PTIL's XML format. And actually, there are actually two different BioPACS standards. There's an older one, which is level 2. There's also a level 1, but that's not published. The level 2, and then the current one is level 3. So you can specify which standard you want. The advantages of retrieving pathway data in BioPACS is that it shares a common ontology with exports from other pathway databases and can use common tools. So, for example, you might be able to import it into some other database that has a BioPACS importer, or there's there's something called PACS tools, which enables you to do various things with data in BioPACS format. So that's one of the advantages. The disadvantage is really that it's not it's a decent match for our pathway tools schema, but not an exact match. So some data gets excluded, or various machinations to get them to fit into to get the schemas to meld together. And then the other main disadvantage is that the object IDs in the BioPACS output, because there's not a one-to-one correspondence between a BioPACS entity and a BioCyc entity, the object IDs are all auto-generated. So every time you issue the request, you'll get different IDs. And the BioPACS data does contain the links to the BioCyc identifiers. So it's all there in the data, but the actual object IDs are going to be different for every request. So those are our data retrieval services. Let me switch gears now to move on to our visualization web services. So a lot of people like if you've browsed around on BioCyc at all, you've seen our pathway diagrams. A lot of people like our pathway diagrams. We put a lot of effort into our code that does automatic layout of pathway diagrams. And so our diagrams are all auto-generated, and they can be generated at different levels of detail automatically. So people want to know how they can get these images. Well, we offer a service where you can download a pathway diagram as a GIF image. So this is an image that can be embedded into other pages, and it's highly customizable. You can use one of our preset detail levels, or you can turn on and off individual components, such as enzyme names, gene names, EC numbers, compound structures, etc. You have some control over the font size and the color scheme. For our linear pathways, they can either be drawn horizontally, vertically, or in this sort of snaking around back and forth layout, what we call our snake layout. And then, so there's a complete list of the customizations available in our document. So the URL that you would use to access such an image, again, it's organism specific, so you would need to specify the org ID. Then it's this diagram only, because it's just the diagram. This is a pathway diagram. You need the object identifier for the pathway that you want to retrieve, and then whatever customization parameters you wish to supply. So here's an example of the same pathway with two different sets of customization parameters. This is the tyrosine biosynthesis pathway. These are the two URLs. I chose this pathway and this set of customizations so that they would all fit on the screen. But so here we're showing structures for the main compounds. That's this M struct. And we've turned off showing enzymes and EC numbers, but we are showing genes. So that's this pathway. And we're using our default snake layout. So it curves around when it gets to the, when it can't all fit in one line, using the width that is our default width. The second one is the same pathway, but we're using one of our preset detail levels, which is one that says show enzyme names, gene names, and EC numbers, and all the side compounds, but don't show structures. And then we're also specifying layout in horizontal way. So same pathway, looks quite different in the two diagrams, but both rather nice diagrams. The other visualization service we provide to download an image is to get a compound structure. There are lots of sources of compound structures on the internet, but here's one more where there's no customization here. You just need to specify the ID of the compound, and you get the structure. The other kinds of visualization services we have are not meant for embedding. What they do is they upload data to our BioCyc pages for some of the various tools that we have on BioCyc that are interactive and where you can upload data to them. Obviously, if you visit BioCyc, we provide interfaces for you to upload data. And I'm talking here about our cellular overview diagram primarily and our pathway pages. And the kinds of data that you most likely want to upload is omics data. But so you can do that interactively by visiting the BioCyc website and using our user interface to upload your omics data, for example. But you can also do this if you're developing a website that has some database of omics data sets and you want to be able to on demand upload them or show them on the cellular overview on a particular pathway, then you would use one of these services. So they enable you to apply highlights to our cellular overview diagram, or to paint omics data on it, or to paint omics data on individual pathway pages, or to generate a small table of small schematic pathway diagrams with or without omics data. So what all these requests do is navigate to complete pages at biocyc.org. They're not embeddable, and you have to use the post method to upload data. So just an idea of what I'm talking about. This is our cellular overview diagram. It contains all the pathways in EcoCyc, organized by class. You can zoom in on it. You can navigate. You can do various highlighting operations. You can overlay omics data on it. This is an example. This is a menu of operations that you can do to it. This is all if you're interacting with it in person through our GUI. So this has a whole set of highlighting operations that you can do here interactively. You can also apply them programmatically by submitting one of these requests. So you submit a request. This is for the cell OV. Again, you have to specify what organism you're looking at. You can say the diagram is zoomable, but it has 4 different semantic zoom levels. And you can specify what is the initial one that it should come in on. By default, it will be the lowest level one, which is this one. And then various operations of what you want to highlight. So things you can highlight genes, enzymes, pathways, compounds, or reactions. And you can do that by either name or ID. And all these, what I'm showing here are the operations that you use. So the preface for G, E, P, C, or R for these object types, and then IDs for name or ID. You can do it by substring, and again, it's the same set of objects with the same syntax there. You can highlight reactions by EC number. You can highlight pathways or reactions by evidence code or curation status. You can highlight genes based on the replicon that they are on. Not relevant to E. coli, but more relevant to something perhaps human, which has multiple chromosomes. And you can highlight genes regulated by some transcription factor. And you can include multiple ones of these. So this is an example. It might be a little bit hard to see the highlighting, but here we're showing we want to highlight two compounds by ID, pyruvate and aspartate. These are compound names. And you can see here it lists that the red ones are pyruvate, the purple ones are aspartate. And I don't know if the colors are coming through, but you can see a bunch of compounds highlighted in red over here and some purple ones over here. This is aspartate. This is an example of highlighting the genes regulated by two different transcription regulators. I forget what they are. I think it's CRP, and I forget what the other one was. But and I actually can't read that if it even says. But again, so it's done one of them in red and the other one in purple. A more common use of this is or a more common use case is when people want to upload omics data. So if you're uploading omics data, in addition to supplying the data file, you need to supply a bunch of other parameters. So the data file is, if you go and interactively upload omics data to our overview diagram, you will get a dialog that will ask for these parameters. So it's the same parameters here. And you'll see information about what the data file has to look like with examples. But basically, the input data file is tab delimited text. The first column should contain names or identifiers, and then there are one or more columns of data. So you need to specify what class. So we need to be able to figure out how to map the objects or the identifiers in the first column to objects in our database. So you need the class parameter, which specifies whether these are genes, proteins, compounds, or reactions, or anything. And then column one is the data columns, and it's a bit misleading because you can have multiple columns here that are all column one, and it's just telling which of the data columns you want to upload. And you can do either a single column number or multiple column numbers, either comma separated or with a dash in between. And then you need to tell us whether this data is relative or absolute. So relative is for ratios, log ratios, things like fold change or comparison to control, that kind of thing. Absolute data are things where the numbers are all greater than or equal to 0. So the scale only goes in one direction instead of both directions. So we're looking at things like intensities, counts, concentrations. And then for relative values, you need to tell us whether it's essentially a 0. For log ratios, that you have a scale centered around 0 where you have positive or negative values. But for other ratios, it's a 1-centered scale. So you have numbers that are either greater than 1 or less than 1, but no negative numbers. So you need to tell us whether your scale is log ratios for on or off for 1-centered scales. And those are the parameters you need to supply when uploading omics data. And you need to supply them by using POST with the multi-part form data so that you can supply the data file. To upload data to the cellular overview, you would use this URL with POST, and then in addition to the parameters describing the data file, you would have this omics equals T to say we're uploading omics data, and you just have to specify what the org ID is. and you get something like this, where everything is colored based on the omics value. This is a histogram of how the omics values are distributed. And you can actually play this as an animation or step through the different columns, because there are multiple columns specified. And you can tweak this in various ways, although you have to do the tweaking interactively once you get to this page. You can also upload your omics data to a pathway diagram, which uses a different URL. You supply all the same omics data parameters using post, plus you use this URL with the org ID. You specify your image as a pathway, and you need to give the pathway ID. You can also specify a color scale here, which is either computed automatically, or you can specify either a 3-color scale, or you can say I want it, use a maximum cutoff. So, for example, if the default is calculated from the full range of the data, but if I have like a couple of very outliers, then it can compress the rest of the scale. So instead, I might want to say use some number as my maximum cutoff and put everything above that in a particular color. So those are the different color options. And then you can say how I want my pop-ups to appear. And this also accepts all the same parameters that control the pathway diagram itself that we saw before in the diagram-only web service. So this is an example of what you would see. This is not an embeddable diagram, though. This actually goes to the web page for the pathway. So this is a pathway, and then you see these pop-ups with the showing the omics data over the course. This is 5 data columns from my data file, columns 5, 6, 7, 8, 9. So that's what you see when you overlay data on a pathway diagram. How am I doing for time? Okay, 10 minutes left. Let me talk about the Smart Table web services. So let me quickly tell you a little bit about Smart Tables, because these, even if you don't want to use the Smart Table web services, if you came here wanting to download all our data and I said, don't do that, use Smart Tables instead, let me give you an introduction to that instead, or than that right now. So Smart Tables are what we call sort of knowledge spreadsheets. It's a way to collect and aggregate some list of interesting database objects, which can be of any type, genes, metabolites, etc., including sequence regions. You can explore them interactively. You can export them in various formats. You can filter and combine them. The really interesting thing you can do them is you can apply transformations to them. So, for example, given a list of genes, you can add a column for what are all the pathways that those genes are in. And so you can build up your Smart Table that way with lots of interesting kinds of information. You can share your Smart Tables either with the public or with specific collaborators, and you can use them as input to omics analyses or enrichment analyses. So let me show you some examples. The way you create, there are 2 main ways to create Smart Tables. You can import them from files and then build them up from there, or you can create them. You can issue a query in BioCyc and you'll get a bunch of query results, and you can say, turn all my query results into a Smart Table. And this is just an example of a Smart Table with genes, the products, and the pathways of the gene. We also have a whole set of special Smart Tables. And this is where you might want to start if you're looking to download our data, because the Smart Tables, given a particular organism, for example, all compounds, all genes, this is the all databases, and it's just all of everything. So you can choose one of these Smart Tables, all pathways, all publications that we reference in this database, etc. So you can start from one of these. You cannot edit the special Smart Tables, but what you would do is you create a copy of the Smart Table, and then you can add various properties or transforms or whatever you want, and then or filter it in various ways and export the result. So I'm just going to throw a couple of Smart Table examples at you. This is an example of a table of compounds with a few different properties that have been added: molecular weight and monoisotopic molecular weight. This is an example of a gene smart table with the accession, left and right position, and the sequence. The kinds of transformations you can do are really powerful. So, for example, starting from a smart table of metabolites, you can transform that into metabolic pathways, and then from the metabolic pathways, you can get all the genes that code for those pathways. And these are just examples. You can start from a smart table of transcription containing just a single transcriptional regulator and transform it to all the genes that it regulates, and then apply a transformation of that to get the regulators of those genes. And so you can create a smart table of nucleotide positions and transform it to the closest set of genes, and then paint those genes on the cellular overview. So there's a lot of things you can do here. So this is an example of, we have a smart table of compounds, we have the reactions of the compounds, pathways of the compounds, and then we have a transformation of to the genes of the pathways. So accessing smart tables via web services. For right now, I mentioned all our other web services. You have to supply these cookies. You actually, for the current website, excuse me, you don't actually, they don't actually use the cookies. You have to supply the user and password. In the next version of the software, you'll have a choice. You can either supply user and password, or you can pass in the cookies. But the most obvious thing that someone might want to do is retrieve an existing smart table using its ID. This only works for the personal smart tables, not for special smart tables, unfortunately. But you can retrieve a smart table in one of three formats: tab separated values, JSON or XML. And it's this incantation with the st_get, and you list the smart table ID and then the format you want. You can also create a smart table using not post, but put. And again, you will supply the data either as tab separated values, JSON, or XML. BioCyc will generate its own identifier for you and return it. And we give some examples. Other services you can do: you can add a transform column to an existing smart table or a property column. You can create a new smart table as a copy of an existing smart table. You can update the name or description, and you can delete rows or columns or entire smart tables. One of the interesting things that you can do is you can apply an enrichment analysis. So what is an enrichment analysis? The goal here is, given a set of genes or metabolites, for example, that came from an omics experiment, you want to understand what this set of genes that perhaps was upregulated in my experiment, what they have in common. So what you would supply is either a file or a smart table, an existing smart table of genes or metabolites, and the output is a new smart table of you would specify what kind of enrichment you want. You can either get a list of pathways. That's the only one that's available for metabolites. But for genes, you can get a list of pathways, which will include pathway classes, or Go terms that are enriched for the input set. And these will be sorted by p-values. And you can there's also, in addition to pathways or Go terms, for databases where you have transcriptional regulators, you can also look for transcriptional regulators that are enriched, or you can say all of the above. So this is just a sample session that I've created, because this is sort of hard to describe what's going on. But I've created a file here of just a small number of genes. This is my command to create the smart table. So I'm using the put command. I'm supplying my user login, and I'm saying create a smart table. My input format is TSV. my organism is E. coli, and these are genes. And I've removed the part where it prompts me for my password, which curl will do. But and what I will get back is the ID of the smart table that got created. I can then say, okay, let's add a property column. Let's add the accession value to this smart table. I pass back in the ID of my smart table. I'm using the stproperty command. This is my smart table ID, and I say I want to add accession 1. And I have to specify the index. So index means I want the accession 1 of the first column. So there's only one column right now, but if there were multiple columns, then I could I might want and there was a different column of genes, I might want the accession of that column. So I do need to specify which column my property applies to. So all it tells me now is that I've added this property column. My request was successful. Let's add a transformation column. I want to add the reactions of the gene. So each of our transformations has an ID. We provide a table that you can see what transformations map to each ID. And again, I want column 0. And it tells me I added it. So then I want to retrieve my smart table and see what it looks like. And I can retrieve it in either tab separated value or JSON or XML, but it's easier to read if it's a tab delimited format. So this contains all the IDs, but these are the IDs of the genes I provided. These are the accessions because I added the accession column. And these are the IDs of all the reactions of the genes. Then I can say, okay, let's run an enrichment analysis on this database or on this data set. And I want to find what pathways are enriched in this set of genes. So I have to say the type is enrichment, because you can also say it can be either enrichment or depletion. If I have a large set of genes, I say, okay, what's missing here? But this is enrichment. And the kind of enrichment analysis I want to do is from genes to pathways. And what I get back is the ID of a new smart table. I'll show you the smart table on the next screen, but right now all I'm getting is the ID. I can delete the smart table. I'm deleting the original smart table that I created here, and it tells me I deleted it. So now I want to run a new enrichment analysis without the intermediate step of creating the smart table. I just want to upload my tab delimited file, and I want to get back my enrichment analysis. So here I'm using the put command again. I have to supply the parameters for the enrichment and also the parameters for the smart table upload. So the class is the genes, the format is TSV, and the org ID. But this is also enrichment, and with enrichment of genes pathways, it gives me back the ID of the smart table that was created. This happens to be an XML format, whereas this one was in JSON format. I'm not quite sure why that decision was made, but so it is. And then I want to fetch my resulting smart table. And here are a list of pathways and pathway classes with the p-values. And then these are the genes from my original smart table that match that particular pathway. And so, we start out with all of them and then get gradually fewer of them, perhaps. And since my original input set was a bunch of genes from the tryptophan biosynthesis pathway, it's no surprise that the top hit here is that we are enriched for the tryptophan biosynthesis pathway, as one might expect. So that pretty much covers all our all our web services. And that, I know that's a lot. So again, I want to point you to our web services documentation file. Also, we have lots of general BioCyc documentation. We have a user guide, which is textual, and we also have a bunch of video tutorials. And if you're having trouble, you can always email us and ask for help. And we try to be good about responding fairly promptly. And that's pretty much it. So I guess, I don't know if there's time for questions or not.
I'll pop back at that. We do have time for questions. If our attendees want to put them either in the chat or the Q&A, I will ask them. To give them some time to get those questions filled in, Is there any particular like warnings or hints for formatting of a query? Is there a specific order to it? Are there any things that you see pop up that should be avoided?
Um, Not for the ones that I've described here. Um, like obviously, as I said, you have to include your login information. But as far as the order of the parameters, they should be fine. There are some things that might need to be URL encoded, perhaps. I don't think many of our IDs actually need to be URL encoded, so I think they're fine. But for one of these services that I did not describe because I didn't want to get into it all, but is where you can issue a query in a general query using our BioVelo query format. There you have to URL encode it, and you do not want to include any other parameters after the query because otherwise they'll be interpreted as part of the query. Um, but but I deliberately didn't cover that one because I don't think anyone uses it and it's pretty hairy. I'm not aware of any other particular issues.
Fair enough. Do you know what's next in the pipeline? Like what's the next tool that we should be looking forward to? Or are you allowed to talk about it? That is.
You mean relevant to web services?
Correct. Yes.
I don't think I don't think we have any thoughts about what to do next. It's possible we might create services to access some of our other omics analysis tools, such as our dashboards of various kinds. We have an omics dashboard. We have a comparative genome dashboard. I don't think we have anything immediate in our roadmap. We're always open to requests. If there's demand for something, we'll implement it, assuming it's something we can do. So yeah.
Where's the best place to send those requests? Is there an email or a FAQ or something?
BioCyc support.
That's easy enough.